Okay, guys, what we're going to do today, we're going to quickly go back through the anatomy we've talked about, make sure everybody understands. Then we have one more little piece of anatomy we haven't quite done yet, and I left it off last week on purpose because I was hoping you'd learn what we talked about before I showed you this extra piece. Okay? Then today we're going to really get into physiology. I need you to participate. So when I ask a question, I need you guys to really think and answer me. That's the only way I can judge whether or not you're really understanding what we're talking about. Okay? This is not a chapter you can just memorize. This is something you have to understand. Okay? All right, overall anatomy. The kidneys are paired left and right. Okay, how do you tell them apart? Right one's lower because the liver's in the way. What is the job of the kidney? Filter the blood to produce urine. Once it leaves the kidney, are you doing anything to the urine? You're just taking it out of the body. From the kidney, it goes through ureters, and we're going to look a little bit closer at the ureter, the bladder, and the urethra, but they're not very fancy. We'll do that at the very end. So it just travels through the ureters, goes to the bladder where it is stored until you feel the urge to urinate, and then expelled through the urethra. Okay? The anatomy of the kidney itself. When you're looking at a kidney, the first thing you need to do, is find your capsule. The fibrous capsule is the outside coating. That's what gives your structure to your kidney. The inside of the kidney is very mushy. Once you find the capsule, you need to distinguish between the cortex and the medulla. The cortex is any of this region out here, right? The medulla is where you find the medullary or the renal pyramids. And the pyramids are these little fan or triangle-shaped structures, right? At the boundary between a medullary pyramid and the cortex, like right here, that is where you make urine. Okay? Anytime the cortex dips down in between the pyramids, we call that a column. Okay? Once we make our urine inside of this pyramid in the nephron, the urine drains out through a minor calyx. When two minor calyces hook up, they form a major calyx. When more than one major calyx starts joining and we have this large collection area, we call this the pelvis. Good. Then the urine flows out the ureter. Okay? The top blood vessel right here is representing the artery. It is smaller, more perfectly circular, and immediately branches into two distinct branches, the segmental arteries. The vein is not perfectly circular shaped. It's going to be bigger, and you don't see the immediate two branches. You just see pieces coming off of the vein. It's not quite as perfect. The pressure doesn't really have much to do with the size. Size has to do with the amount of muscle. You have more muscle inside of an artery than a vein because the artery is the one that has to constrict in order to build pressure. So I guess in a way it is kind of related to pressure, but it's more the muscle amounts. Okay? So are we clear on the structure of a kidney? This is the macro anatomy. This is something we can see with the naked eye. All right? If we go back through the blood flow, I was nice. I told you need to know the blood flow by the time you got here today, right? But I knew that was a lost cause, all right? But you've got to know this, all right? When we follow the aorta and the renal artery into the kidney, the renal artery branches and gets smaller, becomes segmental. Then segmental branches to become interlobar. Where are the interlobar arteries at on this picture? Coming up in between the pyramids, right? So if you see two pyramids beside each other and you see something coming up in between them, that's interlobar. Interlobar branches to become arcuate. Arcuate arches and wraps around the top of the pyramid. So here's the arcuate branching over the top of a pyramid. What are the little bitty ones out here called? Interlobular or cortical radiate arteries. From there, the blood flows through the afferent arteriole into the glomerulus, which is our first really important capillary. We need to understand how it functions. what happens at the glomerulus? Anything small enough filters out and goes into the nephron, right? And then we're going to pick through it and double check in a little while, make sure we only took the bad stuff out, right? After the glomerulus, the blood keeps going into the efferent arteriole. Everybody understands we're not talking about the flow of urine or filtrate or anything like that, right? We're just talking about how the blood gets into the kidney and how the blood gets back out, okay? After the efferent arteriole, the blood vessels branch to become peritubular capillaries or a vasa recta, which is a type of peritubular capillary. What do we do at these capillaries? Double check, and what does that mean? Reabsorption and secretion. That's where we say, wait, this is good. I don't want this to go out in my urine. Let me put it back in the blood. Or, wait, there's something bad in the blood. Let me throw this into the urine because I really want this to go out of my body. That is where we really get the specifics of what we want to go out, what we want to keep. Okay? From there, the veins then get bigger and go out in the opposite direction of the way the arteries came in. Okay? Where does the nephron interact with this blood flow? At the glomerulus, Bowmans capsule wraps around it, right? Remember, Bowmans capsule has the podocytes, the big feet that wraps around it. So it's like a big cup, and all the liquid flowing down flows into Bowmans capsule. Where else? Peritubular capillaries. They wrap around the proximal convoluted tubule, loop of Henle, distal convoluted tubule, collecting duct, which is the next thing we need to look at and make sure you understand. And when I gave you your quiz, the first thing I heard was, this is backwards. Is the human body always going to be one direction? Nah, remember, here's the collecting duct. There's going to be another nephron on this side coming into the collecting duct. I purposely gave you one that was opposite, but then I was nice, and I left the arrows on it. All right, but you didn't even need the arrows. How will you orient yourself when you're looking at a nephron? What's the first thing you need to find? Find the Bowmans capsule because that is the beginning, right? And the convoluted tubules are named by whether they're closer to that start of the Bowmans capsule or further away. Okay? So here's Bowmans capsule wrapped around the glomerulus on the inside. What is this structure together called? Renal corpuscle. Oh, that was your question. Okay. So glomerulus is the blood vessels on the inside. Bowmans capsule is the part of the nephron wrapped around it. The whole thing is the renal corpuscle. Okay? All right. From the Bowmans capsule, what is the name of the first part of the nephron? Proximal convoluted tubule. Then we have the loop of Henle. Which one is first? Descending, so we go down first. Then ascending, come up. Then distal convoluted tubule. Then collecting duct. The collecting duct hooked up to the minor calyx, then major calyx, then pelvis, then ureter, then bladder, then urethra, and the urine can leave our body. When the urine is inside of this nephron, what do we call it? Filtrate. Okay. This is the afferent arteriole. Efferent arteriole becomes peritubular capillaries, and then all these capillaries wrap all around here, interacting with the rest of the nephron. Right here. Afferent goes in, efferent comes out. Correct. Descending always, they're named ascending and descending by the way the filtrate flows through them. So if the filtrate is coming from the PCT, then it has to go down first, so that's descending. Right, if I just cut everything off and showed you the loop, you would never know which was ascending and descending. You have to see how they hook up to know which is descending and which is ascending. Okay? Let's make sure you understand a couple more things, and then we're ready to start new things today. All right. So we said there were three major processes we were going to talk about. Filtration, reabsorption, and secretion, right? Where does filtration occur? At the glomerulus inside of Bowman's capsule. That's where we do our filtration, right? What does the blood look like inside of the afferent arteriole? So before it goes in, the blood is dirty, got lots of waste. It's full, right? It's very concentrated. What do you think the blood looks like in the efferent arteriole? It's got some proteins in it. Does it have anywhere near as much stuff in it as the afferent? So where's the blood pressure going to be higher, in the afferent arteriole or the efferent arteriole? In the afferent, because there's more stuff in a confined space, so the blood pressure is going to be higher. Good. Everybody understands that. All right. The last thing we were doing as we left on Tuesday was looking at just what the type of cells were that compose the different parts of the nephron. Okay? And that's going to really be important as today we're going to go through in much more detail exactly what's going back and forth. So in the PCT, we have cuboidal cells with microvilli. So that means we're going to be doing a lot of absorbing. Anytime you see microvilli, that means increased surface area, so we can really absorb a lot. Loop of Henle, each side is different. The descending limb is thin. The ascending limb is thick. That's going to determine what moves which way. Okay? Distal convoluted tubule and collecting duct do much more secretion. And what we're going to see today, the distal convoluted tubule and the collecting duct, that's where our hormones are going to have a big effect. And after we finish today's lecture, you're going to understand the kidneys really don't know what to do if they don't have hormones telling them when to keep stuff and when to get rid of it. That's really going to be our key control. Okay? Distal convoluted tubule and collecting duct at the end. Okay? All right. So, let's look at our capillary beds. We have the glomerulus. We've already said blood pressure is high in that glomerulus because we had all the stuff coming in. As the blood leaves the glomerulus, blood pressure is lower. Let's take that thought a little further. What do the peritubular capillaries look like? Let's make sure we understand where they are. We have efferent arteriole becomes peritubular capillaries. Is the concentration of the blood in the peritubular capillaries more like the efferent or more like the afferent arteriole? More like the efferent. So what does that mean the blood looks like inside of the peritubular capillaries? It's cleaner, but it's also kind of empty. There's not much in it, right? Remember, we don't only take waste out of the glomerulus. We accidentally take some good stuff out too, right? And what are we going to do with the good stuff? Put it back in the blood. We're going to reabsorb it. Okay, do you see where we're going with this? So the peritubular capillaries, that blood doesn't have much in it. A little preview. Where do things naturally like to go? From high pressure to low pressure or low pressure to high pressure? High pressure to low pressure. Okay? If the peritubular capillaries don't have much in them, what's their pressure like? Low. So things are going to naturally flow from the nephron high to the low peritubular capillaries. So we're going to be pulling things into this peritubular capillary, getting the good stuff back in. Vasa recta is a type of peritubular capillary that we're going to see is specifically involved in concentrating the urine. What determines whether your urine is concentrated or not? The amount of what in it? Water. If I have dilute urine, do I have more or less water in it? More water in my urine. So am I reabsorbing more water or secreting more water? Secreting more water. Does everybody understand that? If I've got more water in my urine, that means the water's coming out, right? So am I reabsorbing it if it's coming out of my body? No, I'm not putting it in my blood. Okay? Good. You guys are predicting things well. All right, the last piece of anatomy we need to cover so we can go over hormones and how things are working today is something called the Juxtaglomerular Apparatus. Sounds horrible, but what does juxta mean? If something is juxtaposed, that means it's beside it. Okay, so juxta means close to or next to. If it's close to the glomerulus, what does that mean? Is next to it. That tells you where this apparatus is sitting. So that's going to be helpful if you can kind of look and predict what words mean. You have a juxtaglomerular apparatus to go with every single nephron. Its job is to regulate what the filtrate ion concentration is, mainly salt. So it is constantly sensing what the salt concentration is inside of your filtrate. Okay? Can you understand it when I say if there's too much salt, that means there's not enough water? Right? If there's not enough salt, that means there's a lot of water. So that's how it looks at salt concentration. Okay? It also helps to watch your blood pressure. Why would blood pressure matter in your kidney? Very good. If you have high blood pressure, can you understand that means it's going to force a lot of blood into that glomerulus? If you've got really high blood pressure in your glomerulus, not only is the stuff we want to come out going to come out, it's going to force all kind of stuff out because you got all this stuff coming and just pushing things through those little filtration slits. Okay? So this is also going to affect our blood pressure, and we're going to look at how. Correct. If your blood pressure is too high, right, absolutely. Okay? The way we can do these two things with the juxtaglomerular apparatus is accomplished because we have two types of cells in the juxtaglomerular apparatus. We have juxtaglomerular cells, these first ones. Their job is to secrete a hormone called renin. And we're going to look very specifically at what renin does for us today. Renin is involved in regulating blood pressure. And we're going to go over specifically how. The other types of cells we have are called the macula densa cells. Their job is to control the density. That's where the word comes from, the density of the filtrate. That means we are controlling how much salt we have in our filtrate. Okay, so that's what we're going to do. This is a picture showing you what it looks like. Okay, so up here we have our nephron. Here's Bowmans Capsule PCT, descending loop of Henle loop, ascending loop of Henle, distal convoluted tubule. The juxtaglomerular apparatus is right here in the center. So if we blow this up, over here we're looking at afferent arteriole coming in. All these little bubbly things are supposed to represent the glomerulus. Okay? This is the efferent arteriole. This is a piece of the nephron coming back up. So the way this looks, have glomerulus, proximal convoluted tubule. Everybody's with me? Loop of Henle, distal convoluted tubule. Okay? So, In a picture like this, you couldn't tell. You would have to be able to see the blood vessels going into it to tell the difference between afferent and efferent. If I just showed you this little piece, you can't tell the difference. Well, yeah, I mean, on this one, right, you have arrows. But if the arrows weren't there, you wouldn't be able to tell the difference between afferent and efferent. Okay? Let's make sure we understand a few things. What does the filtrate look like in the PCT? Let's think about what we just did. We just took everything out of the glomerulus that was small enough and put it in the filtrate. Does it have a lot of stuff in it right here? Yes, it's got a lot of stuff in the filtrate. As we keep going, should we have less stuff in the filtrate? Yes, everybody understands that, right? Okay. The cells over here next to the distal convoluted tubule are the macula densa cells. So they're going to sense the amount of salt in the distal convoluted tubule. All right, let's see how well you guys are keeping up with me. If the macula densa cells predict there's a lot of salt, what's wrong? Okay, so let's think about this. Here comes all this filtrate has been floating through, and it gets all the way over here to the end of the nephron, and there's a lot of salt. What's going on? You've reabsorbed too much water. You're keeping too much of your water. Or maybe you didn't reabsorb your salt. How about this? What if it's flowing through the nephron too fast? If it's going real fast, can you reabsorb enough? No. So these are going to be problems that the macula densa cells can predict simply by looking at, oh, there's too much salt here. That can mean I need to quit reabsorbing the water, I need to start absorbing more salt, or I need to slow down the filtration rate. I need to make it go a little slower. Okay? Good. We're building. It's not supposed to just be popping all of a sudden. It's supposed to slowly be coming together for you. Is that what we're doing? Everything's starting to make sense? All right. The other cells, the juxtaglomerular cells, are in here next to the glomerulus. They sense blood pressure. When they sense a problem with blood pressure, they can secrete vasoconstrictors or vasodilators. What does that mean? They can make the blood vessels bigger or make the blood vessels smaller. What was your question? They're these little bluish purple things all in between. You got to remember the glomerulus is all twisted and wrapped together, and these little juxtaglomerular cells are kind of stuck all in between because they're monitoring the blood pressure throughout. Okay? So let's think about what they're going to do. I didn't that time. Just saying that the glomerulus, we're not really talking about the Bowmans capsule. We're more just looking at what's going on with the glomerulus. Okay? So all these little juxtaglomerular cells are wrapped around the glomerulus. If the blood pressure is really, really high, we've already said that would be bad, right? The blood pressure in the glomerulus is too high. That means we're going to be spitting too much into the nephron, right? Okay? So a little term you're going to see starting to pop up a lot is the GFR. That stands for the glomerular filtration rate. What is a rate? The speed, how fast something is going. Okay? So if the blood pressure is high and a lot of stuff, too much stuff, is leaving the glomerulus and going into the nephron, will you have a higher rate or a lower rate? A higher rate. Good. So high blood pressure leads to a high GFR, a high glomerular filtration rate. All right? What would happen if, since I have a high blood pressure, my little juxtaglomerular cells, they start secreting a vasoconstrictor? Okay? So I've got a bunch of stuff flowing in, and I say, wait a minute, let's close this afferent arteriole. Can as much come through at one time? No, the tube's smaller, right? That makes sense? Now, yes, if the tube is smaller, the amount coming through is going to be coming through forcefully. I agree with you. But we're not trying to control how fast the blood's coming through. We're trying to control how much is coming through. Okay? And that's something very easily confused. I confuse myself all the time with it. Okay? So I'm going to say it again to make sure I'm saying it right. I'm going to say it slow. Okay? High blood pressure. Our GFR is too high. I need less blood to come in here. So the juxtaglomerular cells secrete a vasoconstrictor. I constrict the arteriole, less blood can come in, so now I've lowered the blood pressure in the glomerulus and fixed my glomerular filtration rate. What I'm saying is written on your slides, I promise. I'm just trying to do it with a picture so you can see it. We're just worried about how much space we have to let the blood flow through. Is everybody staying with me? All right, let's do the reverse. Let's say, man, I've been sweating. I've sweated all this water out of my blood. My blood pressure's low. Believe me, that can happen to you. You can sweat too much that you'll lower your blood pressure. So my blood pressure's low. How will low blood pressure affect the glomerular filtration rate? The rate is too slow, so you're absorbing way too much stuff, right? So these juxtaglomerular cells are going to say the blood pressure's too low. I need more blood to come in. to make up for that. How will I get more blood to flow in? I'm going to open up or dilate my afferent arteriole. Now more blood can flow through, so higher blood pressure, get more into my nephron, increase my glomerular filtration rate. Are you guys understanding? Y'all act like you are, and that makes me so happy. Does that make sense? Okay. Let's do the same thing with these macula densa cells over here. So my blood pressure's high. I have a high glomerular filtration rate. Okay, that means, again, flowing through really, really fast, right? If it's flowing really fast, am I going to absorb as much? So what is the stuff, the filtrate, going to look like when it gets over here to my distal convoluted tubule? A lot of salt or not much salt? A lot of salt. Macula densa cells are going to say, whoa, too much salt. So what are they going to want you to do? Slow down the filtration rate. How are they going to get you to slow the filtration rate? Constrict the blood vessel so less flows in, so we get a slower filtration rate. Very good. Okay, everybody seeing how this fits together? Okay. The other thing we can do, besides what we call the myogenic mechanism, just affecting using muscles to close and open blood vessels, is we can secrete hormones. Okay, so I'm going to flip to some of the words because y'all act like y'all really need to see it written. Okay, we've been saying all this stuff. All right, so let's look at how we're going to secrete our hormones. Okay, that's the renin-angiotensin mechanism. All right, the way this works, it's going to involve two things. We have angiotensinogen. That is a protein that's always in your blood. It's just floating around your blood all the time, not doing anything until your juxtaglomerular cells sense something's wrong, the glomerular filtration rate is wrong, and the kidney secretes the renin. When the kidney secretes the renin, that activates angiotensin. Now angiotensin starts working. Okay? Everybody still with me, right? So what does angiotensin do? It does several things. The first thing it does is constrict the arteriolar smooth muscle. It's a fancy way of saying we have constricted the afferent arteriole. And if we constrict it, there's less flowing in. So what's that going to do to our glomerular filtration rate? Bring it down, slow it down. We're letting less salt in, less water in, so we're going to slow the rate down. If we slow the rate down, are we going to absorb more salt and water or less salt and water? I heard both answers. Okay, let's say it again. If we have slowed the glomerular filtration rate, right, it's going slowly through, so can we absorb more or less? More. Think about it this way. If you're standing at a parade and they're throwing you candy, will you catch more if they are going 60 beside you or if they're going 5 as they go through? The slower they go, the more you can get, right? Stupid way of making you remember it, but you'll remember it. Okay, that's my goal. Don't ask that. I'll tell you that after class. Okay, don't worry about that. I should take that off. All right. So far, we are saying we can do that. In addition, that's what we've already talked about, right? That's what I was describing a few minutes ago with the picture. The other thing renin that can do, which is the new thing, is when it activates angiotensin, it causes you, we said we're going to reabsorb more. We do that because our adrenal cortex, where's the adrenal gland at? Sitting on top of the kidney, right? Cortex means outside of those glands. Inside the adrenal gland secretes adrenaline. Outside secretes aldosterone. If you remember back the first day of class, we said adrenal glands make aldosterone, and I think I gave you the definition that aldosterone helps you go to the bathroom less. Well, now you should be able to start thinking about how does that really work. If I secrete aldosterone, aldosterone makes me reabsorb more salt. And here's an important key for any test question you ever take on urinary system. Wherever salt goes, water is following. Salt is the one that starts out everything, and everything else just kind of follows along. So if I reabsorb more salt, that means I'm doing what with my water? Reabsorbing more water. If I reabsorb the water, do I have as much to pee out? Nope, because I'm keeping it. Okay? It's not done yet. Renin says, you know what? Not only am I going to constrict my afferent arteriole to help reabsorb more water and salt, and secrete aldosterone, trigger the secrete of aldosterone, which will make me reabsorb more salt, I'm also going to trigger the hypothalamus to release ADH. What does that stand for? Antidiuretic hormone. Antidiuretic makes you go to the bathroom less by making you absorb more water. Do you see how renin and angiotensin are just making several things happen, but the end goal is still keep the salt, keep the water, go to the bathroom less? See how it's all working together? The angiotensin also activates the thirst center in your hypothalamus. Obviously, you need some water. Your kidneys are trying to keep as much as they can out of the blood, so at the same time, angiotensin tells your brain, tell this dummy to drink some water where I won't have to work so hard. Because the overall goal here is I need water in my body, so don't pee it out. Don't get rid of something that I need. Okay? Now, I wrote it all in words. This is a summary that I wrote for my class last time and accidentally saved it, so I can't make you tell me the steps and type it out with you. But what I am going to tell you is to really get this, I think we need a nice big picture of how it all works together. And I don't have it for you today because it came from another textbook. I'm going to photocopy it and I'm going to bring it to you in class next time. Okay? A picture that shows you how all this works together. But I'm going to give you the scenario that's in the diagram. Okay? Let's say I've been playing basketball and been really going after it and I've sweated. Right? So if I've sweated, I've lost some water, right? Where does the water come from when you sweat? You ever wondered where all that water was coming from while you were sweating? Your cells get it from your blood. You are draining your blood of water when you sweat. So if I have less water in my blood, what's that going to do to my blood pressure? Less water lowers my blood pressure. So this mechanism is going to start out by sensing that low blood pressure. Okay? It doesn't have to be because you were playing basketball and sweating. It could be other reasons, but that was just the example I came up with, right? So I have low blood pressure. These things are separated because this is all going to happen at the same time. So one thing that's going to happen, low blood pressure is sensed by the hypothalamus, secrete antidiuretic hormone. Antidiuretic hormone goes and tells what part of the nephron is affected by hormones? The beginning or the end? The end, distal convoluted tubule collecting duct. Antidiuretic hormone goes down there and tells those, keep water, reabsorb more water. Okay? At the same time, the juxtaglomerular apparatus said, low blood pressure. That means my GFR is too low. So juxtaglomerular apparatus secretes, this isn't in your PowerPoint, if you're sitting there looking, okay? Juxtaglomerular apparatus causes you to secrete renin. Renin activates angiotensin. Angiotensin causes the adrenal gland to secrete aldosterone. Aldosterone goes to what part of the nephron is affected by hormones? DCT and collecting duct, and tells them to reabsorb salt. If we reabsorb salt, what always follows? Water. So the end goal is we have reabsorbed water and raised our blood pressure back up. Okay? The reason you don't have this slide is because usually I tell you, I want you to sit down and see if you can write the steps out. Not necessarily writing this down, I'm not crazy. But that is something you should be able to do. You need to get you a blank sheet of paper and think about it. If I have low blood pressure, how is all this going to work together? See if you can draw the path and figure it out. Okay? Did you guys keep up with this today? This is not complicated, but it's involved. There's a lot of little things working at the same time. So you got to keep it straight in your mind. Okay? All right. So let's look a little bit close. We've been talking about all these things. Let's look a little bit more closely at reabsorption and secretion. When I reabsorb, let's make sure we all understand. Reabsorption, by definition, is moving from the nephron tubules back to the blood. So if I'm reabsorbing, am I trying to keep something or get rid of it? Trying to keep it. In general, do I reabsorb good stuff or bad stuff? Good stuff. There are times when you accidentally reabsorb bad stuff, but is that big of a deal? No, you may be able to secrete it later, or if you've reabsorbed something you didn't need, worst case scenario, it flows back to your heart and it'll make it back to your kidney in a few minutes, and then you can get rid of it again. Think about this. Is it bad if you start urinating out something good? Can you get it back? No, it's gone. Okay? So it's not that big of a deal if you reabsorb something bad. You are going to reabsorb 99% of the things that you filter out in your glomerulus. Okay? And that, I think, is the piece that people have a hard time accepting. Why did I filter all of this out if I'm just going to put it all back in? Because it's a process of trying to ultimately get only the bad stuff in the nephron. And your kidney is not a brain. It can't look at each little thing and go, yep, bad, bad, good, good. It can't do that. All it can do is just pull everything through and use things moving back and forth in an effort to ultimately get it the way that your body needs it to be. Okay? So how do we reabsorb everything? Everything that we reabsorb is linked to sodium reabsorption. So the answer is why. Because sodium movement occurs through every cell membrane in your body, right? A&PII, biology class. What is a sodium potassium ATPase? What is that thing? It's a pump that's moving sodium, potassium, different directions, right? You have them on every cell in your body. That's how you maintain membrane polarity, so you can do things like muscle contractions. Remember we learned the plus on one side, minus on one side of a cell membrane? It's how you do nerve impulses. So you've got these sodium potassium pumps on every cell already. That means if I'm going to move some sodium, if I'm going to reabsorb some sodium back in the blood, I'm already using energy to move sodium. So I'm just going to link everything else to that sodium movement. So sodium moves by what we call primary active transport. What does active mean? Actually doing it, using energy to do it. Primary means sodium is the first one to go, and everything else is just going to jump on and hitch a ride, because you don't want to use any extra energy than what you have to. You don't want to be walking up here and then all of a sudden be tired because you drank a Coke and your kidneys have been working so hard, right? You don't want to be using any extra energy while your kidneys are trying to do something that they're doing over and over again normally through each day. Okay? All right. Well, what else? Everything is going to follow along with the sodium. Your organic nutrients are going to move by secondary active transport. I'm going to show you a picture in just a second. Water is going to move by osmosis, which is the diffusion of water. Okay, so look at this picture, showing you how things are reabsorbed. On the right side, what's your left side of the picture? This yellowy looking area, that's the inside of the nephron. Okay, since we're looking at mostly absorption going on, this is probably the proximal convoluted tubule, because that's the beginning. That's where we really start trying to pull all of our good stuff back in. We don't want any bad stuff, any good stuff leaving. So we try to pull it all back in at the beginning. Okay? Well, if you follow at the top, the first thing that happens, and that's what the red arrows are, is showing you the sodium moving, And the sodium just jumps on this sodium-potassium ATPase. He was already moving sodium. So all the sodium that we need to reabsorb just jumps on. Well, what about all of the other things? Glucose and things like that? It just hitches a ride. So it can actually flow through a transporter protein using the energy from the sodium movement. And it doesn't have to have any energy. That's like explaining, well, I'm going to walk this marathon. Okay, well, I'm going to sit in this wheelchair and you can just push me, right? We both get there and we use the same amount of energy as just one person going by themselves. They do not bind chemically. They just flow through the membrane in a channel that they can only go through if they're both going through at the same time. One of you had a question. You figured it out? Okay, good. I love it when you figure it out. Okay. Whoever sat down and figured all this out, I don't know. I'm glad it wasn't me. All right. Okay, water. See, water is down here by itself, right? Because water just moves by from osmosis, from where is there more water to where is there less water. What did we say the blood pressure looked like in the peritubular capillaries? Low, right? Because we took everything out. Efferent arterial has low blood pressure. Peritubular capillaries have low blood pressure. If blood pressure is low, it's low because we are lacking water, right? So there's obviously going to be more water in the nephron, so it just naturally flows from where there's more to where there's less. Right. Yeah, you do lack the salt and everything else in the blood, but the salt, sodium, potassium, chloride, those ions are charged. That's why you have to have all these channels to help them get an energy in. That's chemistry, trying to understand how they get where they're going. And, I mean, are there other things that go? Absolutely, there are other things moving back and forth, but for the purposes of this, if you can just keep up with what the salts do in the water, then you're doing pretty good. Okay, everything else just kind of comes along. Okay? This is an extremely important slide for you to pay attention to. This sums up the movement of everything through the nephron. We hadn't done all of this yet. So far, we've been looking at the reabsorption. Okay? And if you look, most of the reabsorption happens at the proximal convoluted tubule. So let me draw something real quick. Make sure you understand why that's where we're doing most of the reabsorption. All right. So here's our glomerulus, efferent arteriole. Bowmans capsule wraps around. Proximal convoluted tubule. Loop of Henle. Distal convoluted tubule. Collecting duct. Does that make sense? Okay. All right, let me get a different color, or it's going to all look like black lines drawn together. Peritubular capillaries, right? Make it bigger. All right. So, the blood, right here, efferent arteriole, and the blood in the peritubular capillary. What's the blood look like right here? Very thin, almost empty. What does the filtrate look like right here inside the proximal convoluted tubule? Very concentrated. If things naturally like to go from where there's more to where there's less, does it make sense to you that the major place I'm going to be doing my reabsorption is right here? Because there's a ton of stuff inside of the PCT, so I'm naturally going to let it flow into the peritubular capillaries. This is the site of most of your reabsorption. Okay? It's going to reabsorb and secrete until everything is balanced and we're only getting rid of bad stuff and we've kept all of our good stuff. That's where we're ultimately trying to get to. Okay? So, what we'll be doing next time is looking specifically at what happens in the loop of Henle. And it gets a little complex because one side of the loop of Henle always does the opposite of the other side. So we're going to save that for next time. We have already gone over what ADH and aldosterone do, right? They cause you to reabsorb water and sodium, depending on which hormone we're talking about. And where did we say they worked? Distal convoluted tubule, collecting duct. Okay? See, we're really almost done. right? Only thing we have left to do is look at what the loop of Henle does and then say this is what the ureter does, this is what the bladder does, this is what the urethra does. How are we feeling about the urinary system so far? Okay. Not that bad, right? We've been doing it slow on purpose because I want to make sure you understand. Anytime you get confused, you need to go back to the original, what was the purpose? I'm trying to ultimately get the bad stuff out of the blood, keep the good stuff in the blood. I can't magically pick good and bad, so I bring it all into the nephron and then start putting good stuff back in and double checking and getting it just right. Everybody feeling okay? All right.